everyone. Thanks, Uliana, for the nice introduction and for the possibility of speaking here. Thanks, everyone, for interest and coming by. So I want to spend the next 35 minutes or so on telling you a little bit what we've been doing to understand the model drug perturbations. And I'll be speaking about sort of the type of modality that we use to understand those perturbations is a measurement that we do in single cell, which is called single cell genomics. So essentially doing transcriptome analysis or other types of readout from a single cell. This has been a modality, sort of an analysis, sort of a data set that has been revolutionizing the computational biology field quite a bit, I would say, and so it's been really contributing to many areas of biology. It's going to be big, and that's why we need machine learning for that. So essentially, the method that we're using is many things based on representation learning. I'll show a few examples of that. And in the main part of the talk, I'll be speaking then about how we do this model, this drug perturbation model, which is a collaboration with Facebook AI, which I want to sort of highlight a bit. And then in the end, I show a little bit about resources that you can maybe also use to go into that. If there's questions, I'm happy to take them afterwards. And of course, also happy to discuss via Twitter or something like that. And yeah, I guess I just get started. Since I was expecting that the audience is rather diverse, let me just spend a few slides in explaining what the type of data set is that we are facing. And this is from the area called single cell biology. This is by the way, a nice slide from a review from Alex van Oudenaan. He called it nicely, every cell is special. And in the time, we just used microscope. This is how cells have been found to begin with, right? And then people have been following cells over time and making movies of cells and then putting labels on top of them and trying to understand which compartment cell is doing what. So this is a modality that has been around for a long time, but it's not super high throughput in terms of components of what the cell is. And in contrast, there's this field of omics biology, so to speak, where let's say 5 to 10 years ago, a lot of assays have been around to observe what thousands of genes, the human cell, for example, we have something like 20,000 or so transcripts, different transcripts active. What many of those do at the same time. So complex correlation patterns that could be observed and essentially describe something like the underlying regulatory effects between those different genes. And it's a bit of a holy grail, I would say, in computational biology to really understand those regulatory networks, because then you can perturb and do something on top of it. And today we want to speak about perturbation. Problem though, in that time is that this is just the ensemble averages. So you were usually taking an average over tens of thousands of cells and 100,000 even. So you didn't see an actual state of a cell, but just like the average behavior. And over time, these things were coming together with different types of assays I won't speak about, but with modern single cell RNA sequencing, you can observe now many genes across many cells at the same time. And there's all kinds of extensions nowadays that also add spatial variants, in particular also perturbation variants on top of that. So I think very exciting in terms of modalities. And as so often in computational biology, we're really driven by those experimental assays and building methods to fit on top of that. And I want to focus on these type of high throughput, so high dimensional, but then also many observations, so high n numbers at the same time. And I think I just sort of really want to stress that this power of many is something that has been a game changer. In the early days of these ensemble averages, we usually just had like n numbers, so number of different samples between like, let's say 10 or maybe 50, maybe a bit higher, but earlier it's even just 3 or something. So very low sample numbers, but very high dimensions. So this was in statistics what's called small n large p problem. Whereas in this case now, we're having many at the same time, because this is one of the tricks. Does the movie animate properly? Can you see this? Uliana, I guess it should work. I just assume it does. But essentially what you get here is that there's each of those small droplets are water droplets embedded in oil, and they capture those single cells. So each of those things, some of those you get a single cell, and then you do some analysis that you barcode it, you throw them together. That's why you get many of those observations at the same time. So this is becoming a big data set. And even a few years ago, we already had data sets with more than 1 million of those transcriptomes, very sparse, very noisy, that have been observed. So so data matrices of 20,000 genes times a million or something like that. So this is really a not small n large p, but like a fat matrix type of problem. And for example, condition numbers of those covariance structures really will go strongly going down. So you really came into a range where you could do new types of analysis. That's why I think the field has been picking up many kinds of machine learning ideas in that. This has been realized by the field in Nature Methods, biggest method journal, like I would say, around, has been denoting this already in 2013 as method of the year, and has been following it up with 2019's multimodal, so different things, not only RNA, but also protein or TAC or other things, chromatin, observed at the same time. And then the transcriptomic one, also the spatially resolved one, also method of the year. So 3 times in within 6 or 7 years become a method of the year, of course, shows you how important this thing has been and how many people have been taking it up. And for that, that you produce all these complex data, you need tools to analyze and do something like that. So essentially learning, now that you can quantify such a position of a cell in this high dimensional transcript space, essentially across its variability, can tell you about decisions. This is from a recent review that we did. Essentially, you see that healthy cells could be making some type of trajectory, say natural healthy aging, but could take wrong turns and could turn to all kinds of diseases. So if you zoom in there, essentially what could potentially happen, as you sort of visually indicate here, is that maybe a cell could take wrong turn to one of the other cancer. And ideally, you find what's going wrong there in terms of transcript, you perturb it, say with a drug, and you put the cells back on the healthy trajectory. So this idea that diseases are originating obviously due to cellular processes, and if you understand what cells go through in terms of trajectories, you can then actually sort of use this to change disease processes, I think is quite big. So what's the machine learning problem behind it? Well, the machine learning problem behind it is essentially we want to learn trajectories, right? So we typically lose the cell at whatever type of observation we do, so we don't see cells resolved across time. But we want to understand the trajectories behind it. So this has been a big question in the field, and contributions we've been doing here, and this is now just one for now, not for those omics, but just for images because it's easier to visualize. Here we've been looking at cells going through a process, which is important in the core of cell biology, which is called mitosis, right? So it's just going through cell division. And we had a machine that was taking essentially many pictures of many of those cells, just morphometric pictures, in addition to some colors that told us about the different cell cycle states. So we had a label for those. And the question we had, can we predict based on those images which cell cycle state they're in? In the first paper, we generated some handcrafted features, such as morphometry, such as roundness, texture, and so on. But in the follow-up paper, we've been just throwing this into a plain old CNN with some small modifications, and then visualized the last layer of this network, the dimension reduction phase, and obviously added a classification loss. And with this, we got fantastic accuracies for predicting the particular cell cycle state. And this is what you see here. We also learned about the process. So here you see the network in the last layer that has been trying to classify those different cell cycle states. But so far, nothing has been learned. So it's just a big messed up ball. But each of those labels are now being trained, and you see now the network going through the different epochs, how it starts updating this particular separation of those cells. And what you can start hopefully appreciating is that those different cell cycle states really start separating across the different categories as you would like to have, because you need to classify it in the end. So it was this stupid neural network was really learning this complex biological process. And there's something more. For example, if you zoom in here, in this case, this has been a doublet. This has been something that should have been moved away by the machine in terms of pre-processing. This was automatically put in a separate category. So even though we didn't tell it about it, the network was learning about outlier type of processes. So this is a very nice model to really describe the variation in this cell data set in terms of morphometry. So we learned this dynamic process. We just had a classification problem, but we learned something continuous. And that's really something that has been very important for all kinds of downstream applications. And I'm going to be talking about the omics in a minute, but we've been using this also for human classification. So yeah, we won we went into winning ranks or sometimes some type of data bowls with these type of approaches. But we also transported this to human, in this case, for example, retina observations. Each of those dots now is not a single cell, but a human person. In this case, having a particular disease, diabetic retinopathy. And then we were ordering, even though we didn't have a time resolved observation, but just like different people, we were ordering them according to similarity with respect to this classification, and we learned about different cell cycle, about different stages of that particular disease. And nicely, you know, the medium state, for example, was between the severe and the mild, when the mild was closest to the healthy. So you could hypothesize that there's actually trajectories also happening here. We're actually following this up now with a complex data set with a local clinic. But let's get back to the cell. So we learned about trajectories. So this trajectory learning is something that's actually, I think, quite big in the field. And what you would do now with the single-cell transcriptome is essentially you could ask questions. So for example, you could have cells that are in a healthy situation, and in a pathological situation, you have a bunch of cells that do something different. But if you were just taking averages, you wouldn't pick it up. But if you do the single-cell sequencing, you do some clustering of the profiles, and you do dimension reduction, you maybe pick up those cells that have similar processes or genes expressed that are living close by. And in the pathological situation, maybe a new cell type is popping up that would be then disease associated. And if you were just averaging across all and just take a single number, you wouldn't find them. But here now you would be sort of separating them out, and you can learn something about the disease. And this looks so visual, but actually doing this involves all kinds of computational problems. So people were really coming up again with new dimension reduction techniques, with new clustering techniques, with new techniques for differentially comparing these things. You know, just not taking averages, but looking at variances, things like that. So this has been really driving a lot of developments in machine learning and compile, for example, in terms of manifold learning, that you don't only look for clusters, but maybe also because there might be sort of substructure there, then these trajectories would be, for example. What my lab has contributed next to all the analysis tools is also frameworks for sort of quick compiling or putting together of those analysis, and the ScanPy tool has become one of the standards in the field, downloaded a few 100,000 times, and people really building upon it and really scaled and helped sort of contribute to this analysis. So what would be a machine learning problem that could be asked on top of this? I mentioned this is essentially a representation learning type of problem. You want to see where there's different clusters and so on, but how would you want to be doing this? So the goal is you want to learn biological trajectories from many cells that are doing some types of transitions. So let's say we have those gene expression patterns, for example, and we're embedding them in this high dimension transcript space, and potentially they just form this big old blob. And if that's the case, so I said it's the power of many, so we have many of those, so we can learn something about the structure, but we hope that this doesn't look like that, but there's maybe some substructure there. Cells are maybe here in terms of a stem cell, and then they're making decisions, they turn it to one or the other cell type or something like that. There's been many of those, stories or pictures that biologists have been using to describe how stem cells make decisions. I mean, after all, it's really fascinating that all of us originate from a single fertilized egg cell. I mean, how is that happening? How do these different cell types that we have in our body form? One of the pictures that people have is that there's like a ball sitting at the top of a mountain that rolls down and makes decisions and sort of ends up in the different valleys that would be different cell types. And I think with these transcriptomes, we can approximate this a bit. So one machine learning question you could now ask, if we have C cells during transition, then we can learn about those ridges or valleys or something like that. Here's an example. This is a collaboration that we did with a colleague from Cambridge, UK, where we've been if you just do cluster of those cell profiles, we do see some structure, but not really much. But if we instead now let random walks run along this high dimensional transcript space, we would be essentially going along the manifold, right? If you do many random walks, average across those, you would be basically getting some type of diffusion distance. If we do so, you can now order those cells according to similarity in terms of their distances. This is called pseudo-temporal ordering in the field. Tries to basically arrange cells according to the similarity. And earlier algorithms such as Monocle or beautifully named Vandalus from Pear Lab, and then our diffusion pseudotime have been really been impactful, and many people have been following up. There's more than 70 or 80 algorithms now for these type of ideas. But essentially, you learn now sausages, trajectories, trees, more complex things in the data set. And if you now order those cells according to this distance, if you decide to start somewhere, if you apply this, for example, to this matrix here, what you would get is then a sequence of turnings on or off of those different programs, and it's sort of much easier to interpret. So you still get the same cluster, but you see a branching event here, so you can really make more out of this information. So going beyond clustering, this unsupervised analysis with respect to ordering has become very popular. And I guess from a machine learning perspective, you would call it one-dimensional manifold learning. So it's really the lowest dimensional. Well, it's not zero dimensional, so it's not clusters, but it goes beyond that. This has become quite important, and understanding them more and finding more of those cell types has become a big international endeavor with many people contributing to various efforts. But this one, the Human Cell Atlas, as well as some other ones where I'm also involved, have become one of sort of those flagships to show around. And the Human Cell Atlas, essentially, we try to generate this type of periodic system of elements, not for elements, but for cell types. We really want to describe all the variation of cell types that you have in a healthy body, and then you can sort of apply them for different things. For example, my lab has been doing this for the lung in terms of analysis with experimental partners quite a bit. We've been also leveraging these type of atlases for important recent questions. For example, we've been building a COVID lung atlas and have been trying to understand how these variations, each of those lines now being a human, either measured with whole lung or some type of sub samples, and trying to see which parts, for example, those entry genes from SARS-CoV-2 would be active. Thereby you were able to localize that this is not every cell type in the lung is actually subject to that virus, but just a few of them. And this actually correlates with covariates such as age of the donors. But I want to speak more about the machine learning aspect of it. And while many have been using these graph-based approaches that I was showing before, essentially, letting random walks run across the thing, we've been more recently putting a lot of effort and time into using neural networks for latent-based learning, which is obviously the de facto standard in computer vision and other fields. And we've been thinking, could we do something like that in other fields as well? Idea, because here we don't have labels so much, right? For each single cell, we just observe many single cells, but we don't actually have them labeled. We just have a bunch of them labeled. It's more like a multi-instance learning thing. So instead, we decided, and some other labs as well, of course, to use unsupervised learning techniques such as these autoencoders. Autoencoder is nothing more than an input layer in a network that sort of squeezes things down, and what's then called a bottleneck layer, blows them up again and makes a loss function so the output as similar as possible to the input. If everything was linear, right, this would be just giving you a principal component analysis, but in the nonlinear case, you sort of learn and some of the structures here, and it's actually quite interesting what you can do. For example, you can take gene by cell matrix here that you would feed through, that would be, for example, going from a progenitor towards a differentiated case, such as what we had before. And if you now do then this reconstruction here, it will be filling up the noise. It's like a denoising technique, like just as PCA would be doing, just a nonlinear version of that essentially. And you can then reconstruct things. What's more interesting though, and we've been writing papers along this lines, but what's I think more interesting is you can actually also analyze and look at this bottleneck layer, what it does. And I think this is really fun. So let me just visualize what we've been doing. So there's been quite a bunch of methods to do so. What we've been actually thinking about is to adapt here the noise function for the bottleneck layer to this particular problem. In our case, we do know that we count transcripts. So an MSE loss might not be the best. Instead, we developed what we call deep count autoencoder. This was work by Lukas Simon, postdoc in the lab at the time, now professor in Houston, and Gökçen Arslan, who moved on to the broad and then recently to GeneTech. They replaced this cost function by what's called a zero inflated negative binomial, but essentially some type of loss that extends a Poisson type of count function, and that we just adapted here in the output layer. And with this noise function, we could actually show that we can denoise ground truth much better than a mean squared error type of cost function. And in particular to other type of denoising methods, ours scaled much better with the number of cells, because it's a neural network, right? So they really know how to deal very efficiently, more or less even in linear fashion, with the number of samples. So that was nice. What the main take home for me is, and that gets us closer to this drug perturbation story, is in this bottleneck layer here, you actually learn interesting structure. So this bottleneck layer, in this case, we drove it to the extreme and just made a two-dimensional bottleneck. Each of those colors would indicate a different cell type. Well, you do actually find them in this bottleneck layer. So it gives you a low-dimensional visualization of that. It needs to, right? Because it needs to somehow represent the data very efficiently. So it's like a coding problem. So we've been thinking what we can do with those learned manifolds in the bottleneck. And one of these first ideas that we've been having is we try to copy the idea that domain adaptation and style transfer has been doing rather popularly for generative neural networks in imaging. This case from example with adversarial networks, where people have been sort of trying to represent men with glasses in a latent space, and then doing some arithmetics on top of that. For example, subtracting men without glasses, again in latent space, adding women without glasses, and actually generating samples from women with glasses. It's kind of cool, right? And it's really nice that if you do this actually in pixel space, this wouldn't work out. because obviously the ratio of nodes to I and something like that is changing for each of these things, and just in latent space it would be sort of so regularized that it could be actually working out. So we've been thinking, what can we do? Can we do the same thing with cells? What would be like a perturbation there? What would be these type of different classes? Well, it would be the cell is, for example, stimulated versus a non-stimulated cell. And if we know how the stimulus acts there, maybe we can transfer that stimulus to this unperturbed cell here and generate this one. We either generate these samples because we don't have enough time to do, or maybe understand why it's driving that type of perturbation. So predicting these phenotypic perturbations, I think, would be quite important. For this is work by Mo, senior PhD in the lab, and Alex postdoc moved on to a company. We've been thinking about how to make a generative neural network model to predict single cell perturbation effects. So that's our first perturbation model. This was published 2 years ago. And essentially what we've been doing, we take gene expression space, we have cells responding to perturbation, and we find multiple of those, and then we ask about this new one. What we do is we encode it, in this case with that. With actually a variational autoencoder, so we able to sample from that. And then we hope, just as what was done in the computer vision example I showed you before, that in latent space we can apply arithmetics. So we can hope that there's one dimension where actually this shift, this perturbation effect, is consistent across each of them, even though here it might not be because there's different setups and cells always behave differently in terms of their internal setup to perturbations. Here it might have been straightened out, we apply it and we just go back. So this scGen is nothing more than a rational autoencoder combined with latent space vector arithmetics. And then we're actually able to do rather robust OOD, so out of sample prediction. And this is a hard problem, right? But with this assumption, this was actually working out well, but it just worked for a single perturbation. And here an example, we've been doing this for PBMCs, because you see different colors, different subsets of blood cells essentially under control and under stimulation. And then we left out one of the stimulated ones, and we're actually able to predict that stimulus. So this is very nice. We had all kinds of applications for cross-study integration, for batch effect removal, and even integrating samples across different species. But the limitation was this is a rather rigid model, right? So here everything is constrained to be this linear in latent space and kind of empirical, and also obviously only a single perturbation. So what we now try to do, and this is the idea of our drug perturbation model. We want to have an interpretable model, but also a scalable model for these perturbations. So we try to understand the impact of variation, and then have also the OOD prediction. There's been a bunch of popular approaches to do so. I think one of the most popular ones for sure would be just doing linear models, matrix factorization, linear factors, and so on. If you do that, then you can interpret what's going on. So it's a useful approach, and it can sort of assess the contributions of particular biological factors. But of course, you would assume that at each point in that cell type space or that manifold, the perturbation effect would be the same, which is a strong simplification, which is just not true, right? But we had an application for that. We actually wrote a paper where we've been putting together this collaboration with Stiegler and Marioni lab, cell cycle genes, and we're able to remove them from the thing. And this is a review from Regev and Yosef's labs, where they've been thinking about factorization of gene expression matrices by a bunch of factors that are known or unknown, technical and hidden technical covariates, and all those loadings would tell you what's going on. Well, this assumes linearity, and we just saw linearity is not the best job. The second thing we of course want also for our model is that it's scalable. So you don't want it only to answer for single drug, but for multiple drugs. And combinatorial, like the drug screens is something that the field has been doing. I think we've all learned that during the past 2 years, drug or screen for vaccination. But the second thing, of course, is that in particular in cancer, but in many other situations as well, you want to have combination treatment. So you just want to you see that an effect, maybe not additive. If you combine 2 drugs, you get maybe better effect. Say if you have a screen by 100 drugs, with a single dose, maybe if you just increase the number of doses, you already increase the complexity by sort of an order of magnitude. But if you increase the number of combinations that you want to do, you quickly get into a regime of number of combinations that's just much too much to actually really do that type of experiment. So you need to have some type of computational way of interpolating these type of big matrices here in terms of the combination. So you want to have something that's interpretable and scalable. And our idea was now that we combine the interpretability of linear models with the flexibility of a deep representation. So essentially, we're doing like these factor models that I showed you before, but we do them in this nonlinear latent space. And we need to learn then the loadings in this nonlinear space. So that's the idea of what we call compositional perturbation autoencoder. I should mention this is work by Mo, Carlo, and Yuga in collaboration with Anna Dimovskaya and David Lopez Paz from Facebook AI, who have been strongly pushing for this project. The idea is that again we have this encoder decoder type of architecture, but now we add a discriminator. Let me just guide you through this movie, because this is sort of how Facebook AI also communicates. I really enjoy that type of collaboration. So essentially, we take a cell, we encode it just as we did before, then we add a discriminator that removes now the impact of this particular cell type as well as a drug from that cell, and you get sort of basis cell state that is then added back again and decoded to give you gene expression again. And then you have your loss function that essentially adds this discriminator loss to the reconstruction loss. If you do that, then in the actual testing phase, you can now ask counterfactual questions. You can, for example, ask, how will a cell A behave under drug perturbations of cell B? For that, you just take the cell, encode it, go in the bottleneck, get its basal cell state, so remove its contribution of the particular cell type and drugs, then add a cell type of cell B, add the attribute, and then you reconstruct it. So in this case, you can basically dream up or come up with new type of perturbation effects that you haven't been seeing before. So to be a bit more precise, what the thing does, it's an autoencoder that does sort of potentially interesting things in this bottleneck layer. What it does is it not only takes the latent bottleneck, but it modifies the bottleneck by adding a linear model in the latent space. So by adding essentially for each perturbation, a strength factor here, times a vector that represents the effect of that perturbation. And this vector here, of course, needs to be trained then as well. So basically the parameters that you'd have is then, of course, the encoder and decoder, as well as the coordinates of these type of directions of the drug effect. That's going to be learned across all. So those perturbation effects, those perturbation embeddings, you would be learning via that joint cost function. And now the key trick is, in order for this to work, that if you just left the Z as it was, it could just basically just put all the information here. So you need to get to this basis state. So we added an adversarial loss that was trying to basically predict based on that only the perturbation state. In this case, we removed that type of information here. And in this removed information, so the encoder was made to basically not be able to tell about that, that was just added then here. So in this case, we ensured that this was really pointing in the full direction of the data space, okay? So that's sort of one of the key things. And obviously it's a bit of a tricky thing to be able to train this robustly, but that was the general idea. And then of course you add some nonlinearities here, and we also added covariates such as cell line, which one this has been observed, or some patient information, and we had the same type of training. But what can you do with these things? Well, what you could do is now, of course, you can, now that you actually model each individual contribution and some of the combinations, you can also predict novel combinations. You can also do something like dose response modeling. So for example, if you observe here a model for the dosages at some time points, you can actually interpolate the rest. And I think that's really interesting. This really is interpretable in the sense that those vectors, so these embeddings here that you've been learning, you can actually make sense out of them, right? So you can look in this perturbation latent space and see which drugs are maybe having a similar mode of action, which cell types or covariates are close by. So that's the idea of the CPA model. bioRxiv is available here, but let me show you one or two examples of what this can do before I wrap up. I think I have something like 8 minutes left, so right. Please stop me if I'm going too long. So we've been looking into data sets, what we can do with this. For example, here we've been trying to learn the latent response from a really large scale single cell screen. In this case, this was a data set called Cyplex from Trapnell Lab, who had all kinds of tricks to essentially give you many different cells under. I mean, that's sort of a specific type of architecture he's been doing. Essentially, what we had a data set is with three cancer cell lines across 180 drugs, 4 doses, and 2 replicates. So more than 4,000 perturbations and more than 500,000 of those cells. And you see, we just throw them together. It's like salt and pepper. So all these processes, these effects of those drugs will be somewhat similar. So we've been training the model. And what we can first ask is, does the model interpolate existing data well? In this case, for example, we've been we held out prokinostat effect in this particular cell line called MCF, and then we look how a few genes actually are expressed. And this is the control, and you see that the true gene expressions are shifted, and you see that our predictions actually match up with the true data sets rather well. They're normal because we have a very simple noise model here, but the means and the strength is very strong, and we can more systematically check how the means behave across all transcripts, not only those 5 that I was showing over there. And you see we get fantastic matchups with very high R-squares across all genes, but also for the differentially expressed ones across the different dosages. You can ask how this works then across the different cell lines, or how it works under particular dosages. What I would then, I just want to say here, we did some type of analysis with respect to splits. I'll skip that, but just show you that you can interpret that space. So you can now look at the latent space of the cell lines. That's kind of boring, right? So they're just simply maximally orthogonal. So there wasn't really any shared structure between the different cell lines, but you can do a kernel PCR UMAP of the drug latent space as well. And that's actually quite cool. So in this case, basically, we had those 188 conditions or so, and we can see how similar effects of those conditions were. This we get by looking at the embedding vectors, right? And just doing a low dimensional visualization of that. And what you see is that, in fact, the effect of drugs that target epigenetic regulation, they've been sort of thrown together more or less. And there's been sort of a few others that are sort of grouping up in some of these areas. So you can really try to now see what the drug latent space is doing. And then you can actually also have price and so on top of it. And for example, here we saw that in the dose response, actually the epigenetic regulation ones were sort of given the strongest contribution. The second example, and then I will try to wrap up, is that we've been not only looking at drug perturbations, but we've been also trying to understand genetic perturbations. That's sort of a bit more targeted perturbation, or target drug perturbation if you want. So in this case, we've been essentially doing a CRISPR experiment, or we've been using data from a CRISPR experiment that was just really knocking down one particular gene and also 2 genes at the same time, so really learning interactions. In this case, this is from the Weissman lab. They've been doing a perturb-seq type of experiment that really screened for a particular set of doubles. In this case, they've been trying to understand how genes essentially interact. So what we had is more than 250 single, but then also double knockouts across 110 cells, actually knockdowns. And you can again look at this perturbation latent space. And we know now, because this is now in contrast to a drug, really very specific which gene is doing something. So we know which gene programs have to be targeted. And what we see is that those target gene programs actually very often coincide, which is beautiful. And we also see that we can really model those double perturbations very robustly and kind of really sort of put them in the same place. So this seems to be working out very well. And we really do good interaction pattern prediction. So we can see how the true data matches up with our predictions robustly across different settings. And this essentially, there's some nice bio in there with respect to the types of interactions, which I will skip. But I just want to say now, in this case, we had, I think, something like 5,000 or so potential interactions, but we only observed like 100 or so of those combinations. But because we have this model, we can now fill up all the rest of those 5,000 perturbations with our model. Obviously, it won't be perfect. There might be all kinds of errors, but we see that we have some measures of those effects in those unknown ones, and the rest we're just generating with our generative model. And we have an uncertainty model on top of that, in this case, just cosine similarity in that expression space. But then we can see which genes are actually prevalent in this particular class. And we can basically give biologists now a really full exploration of this landscape in a noisy fashion, with noise and so on, but with uncertainty. But of course, this can be, for example, now used for experimental design. And now that you have this, you can, for example, zone in into one particular interaction. Let's say you're interested in this KLF1, and you actually haven't measured this across all the different transcripts. But now we can just impute it, and we can see that some of those things where we actually have a true measured observation do match up, and some others. I think I stop here. I had this one point about a model zoo that we've been generating to actually give a resource for doing this type of analysis. I just show one slide for that, but then I will stop. So if you're really interested in this and you're sort of looking for data sets to try your most fancy recent representation learning method out, you can just download a single data set, or you can go to this resource which we call Sphere, which is Greek for sphere, And then you can actually combine, you can really download a lot of these data sets, do analysis on top of them, and then also download and share model parameters. So we have really very nice resource for people to really look across human and mouse, across more than 150 data sets each, with more than 3 million cells already contained there and currently strongly growing. So I think it's going to be a fun resource for people to play around and see, what has been annotated, actually very quickly access this type of data. So that's FIRA for you. I think if you want to benchmark things, it's very useful. With this, let me wrap up. I showed you that representation learning is something that's very useful for single-cell RNA sequencing, and you can use essentially these bottleneck layers to also then add perturbations and learn about combinations of that. Just as an outlook, I mentioned that spatial transcriptomics is becoming a very important sort of new direction. And we've been developing tool chains as well as analysis. Call this one thing SquidPy, which is a spatial analysis framework, but then we've been also using graph neural networks to sort of really analyze those more complex downstream patterns. I think it's going to be fun to see where the field is going, also with adding this spatial variant to that. With this, let me wrap. This is how Bavaria looks all the time. This was in a still socially distanced lab retreat. If you're interested in learning more about that, contact me on Twitter or via email. We always have positions open, obviously, and I thank you very much for your attention.